Howdy, my name is Hanna. I'm an associate professor at North Dakota State University in the Department of Animal Sciences. I'll be talking today about data science and biostatistics in the ag and natural resource areas. So today I'm going to talk to you about data science and biostatistics, terms that sometimes make people run away. But I have found through my career that I am more and more pulled to this area, and so I think it's really integral for agriculture and natural resources to consider how these interplay with what we do in research as well as application. I'll preface that I don't have a lot of slides, okay? Mostly because I think the points sell themselves and we can talk about some examples, and then surely if you have questions or want to know perspectives, we can go through that, right, throughout the presentation. So feel free to ask questions as we go. So just a little bit about me in terms of where I come from. I did 3 degrees at Texas A&M University, starting with my bachelor's and then going into my master's and PhD in what they called animal breeding. Fun fact, that's really just quantitative genetics applied into livestock production. All right, so I am a quantitative geneticist by training. I then finished my PhD and went straight to NDSU in a 70% teaching, 30% research appointment. And I've really found a lot of fun going through that experience of becoming a better teacher and mentor while also getting to have fun with research. Currently, my position is 60% teaching and 40% research. And as you can see with my research kind of focuses, I'm really interested in that intersection of how we collect phenotypes, how we use it, and what that means long-term for selection and application purposes. Now, before we go further, I put this image up here because this is really how I got started in my graduate program. We were working with a population at Texas A&M that were Nelore Angus crosses. It was a very structured population. They had created F1s, the first crosses, then they had made F2s, trying to understand segregation on many different concepts. One of the things they were not expecting was to find coat color variation out of what was expected, okay? And so if you know much about cattle, black is dominant, right? And so if you have a black allele, you should have a black animal. There should not be any ifs, ands, or buts, right? But we were finding in a population of cattle that all of these pictured here were the black allele with the wild type allele. So why in the world were we getting partially to almost completely red individuals, right? And now we could have fun with coat color. It's a great visual representation of Mendelian inheritance and some of those things. But what was really driving some of this question is, what if this starts happening in a real producer's herd? Is it happening, right? And how kind of economically would that then impact them if they're trying to certify their animals for different programs like certified Angus beef, okay? So there were some economic implications. My focus was, how do we characterize it? And can we identify any genomic regions that actually lead to this reddening type attribute, okay? And so it was my first kind of endeavor into problem solving that question, placing out how we can use the data that we had or additional data we could collect and trying to figure out what it could be, okay? And so in this whole endeavor, which is now published, we did find that there was a region on the chromosome 6 in cattle that acted like a major gene, right? An on-off switch of sorts. And it was recessive in nature, okay? Then they went on to study, after I finished my master's, I went into more of a genomic evaluation methodology. And what they did find later on with another master's student is that the gray locus falls almost in exactly the same place, okay? And so through that type of topics, we were able to deduce that there's evidence that this may actually just be what gray looks like on a black background, right? Which is kind of cool. So it's kind of my first foray into data science, and I don't even know if I really realized that it was data science at the time, okay? So just to get a feel for the audience, do I have any undergraduates in here? Yeah, okay, great. Graduate students? All right. Faculty and staff? Great. Shout out some areas of focus that you have. Could be cattle, it could be wildlife, it could be birds, okay? I can always count on you. Okay. What other? We got birds.
I got some goats too.
Goats? Okay. Pollinators, awesome. okay? So it's this whole concept, right, of I'm probably actually doing this, and you'll see me point that out a few times. It just depends on how extensive you're really doing some of this data science type attributes. So what I'd like to talk about today is what is data science, and then also what is biostatistics, okay, which is another integral part of your research. And then what kind of application are we seeing? I'm going to use current as kind of my research examples of what's going on, but then maybe open up for discussion of how that's probably trending or going forward, right, with the different fields. So what is data science? It's actually interdisciplinary, okay? And we're trying to aim at extracting or extrapolating knowledge and insights from potentially noisy, structured, or unstructured data. So what is the concept of structured, right? How many of you would potentially classify your data as structured if you have data? Yeah, not many. How many would structure it or call it unstructured? Okay. So really when we talk about experimental design, right, we talk about structure in the sense, can we have replication? Are they fairly balanced? And are we accounting for the other variables that could potentially drive noise in that collection, right? The less that you account for that, the noisier, messier that data gets, okay? And we have to either be able to account for it, or we have to set it aside as its own little entity to analyze, okay? So try to think through that, right? If you've got fairly unstructured data, there's probably other attributes you need to be mindful of. And we can do some of that through areas such as computational science. Pointer works, yes. But we can also do a whole host of things in data analysis called data mining, processing, and visualization. Some of that could be really rudimentary or exploratory in basis. But we could also think about this on the end where we're trying to extrapolate out. We have our basis of our scientific methods, right? How we're going to collect the data, why we're asking the question, and how we actually get at the question. And then we have the statistical side of getting at that question, right? And then we have this whole concept of domain knowledge, right? And I put cattle there because that's been my primary focus. It doesn't mean I haven't dabbled in other species, but it's been where I've primarily focused. So we think about the scientific method, y'all are all probably really familiar with that, right? It's something that goes around with, we take observations in to try to understand a question that we have. We create a hypothesis that potentially we can answer, either by saying we can reject that null hypothesis or we fail to reject, right? Either or. We do a full-on experiment to try to answer that question. We analyze it, and we make conclusions based on what we could from the data that was there. Now, statistics is going to be that area that looks at that experimental design. It incorporates some of that scientific method in terms of data collection. It also does some analysis, interpretation, and then presentation of it for those conclusions. And there's actually quite some controversy on whether statistics and data science are really different, okay? But in reality, if we think about what data science is today, right, it really is a step above what statistics is. Because it's taking on more and more attributes of how we can interpret data. So what is this whole aspect of computational science, right? We often think about that in the sense of using high performance computing or an HPC. I didn't ask this prior to now, but is there an HPC that you have access to, either at Tarleton or through another university? Probably should have asked that. I know Texas A&M has one. I just didn't know if there was access or potential to use that. So NDSU has a HPC that they call CCAST, okay? And CCAST stands for Center for Computationally Assisted Science and Technology. And they have it in two forms. They have it in a clear on, you're going to code, go in, you can't see anything besides just submitting batch jobs to run something. And essentially what you're trying to do is this high level of computing because you need so much RAM, you need so much memory to process the data that you have, okay? But there are movements now to be more user-friendly, and so what we see is that user-friendly motion is what they call on-demand. And so essentially, how many of you have worked in R or R Studio? Okay, so some of you. It's a software that you can do statistical analyses and it's somewhat robust on a regular laptop, but if you're still talking about high level, then maybe you need help from an HPC. And so in that case, the on-demand creates a window in which you can actually use a high-performance computer on your desktop in a very GUI-friendly, the way it looks is very similar to what you use on your laptop, okay? So that's kind of nice, and there's different ways that you can do that. There's different software that they may have available. This is just an example of what CCAST offers right now. Jupyter is a pretty big one for Python. And so we can get into several of these things based on what you're working with. The whole premise for computational science is that we have a large data set we need to manipulate, we need to get information out of, and so we're going to use that big computer to process it. I have toured several HPCs because it's just interesting to see where things are going, because you're sitting literally across campus, sending your stuff to this unknown blank space, right? And you're trying to figure out what it's doing. And when you walk in, it's just this huge room with big fans and big servers, okay? Because there's so much heat generated from those servers that it's just running constantly, right? And then it also goes into, well, how do they back up the data that's running? How do they query jobs and know who gets priority over others? Because there's other people that may want to use it. And so it can be a little intimidating to take that on. And I think that's one of the big things that keeps a lot of people from really being captured as a data scientist, is they may or may not be using this next level of computational science, okay? But other than that, I think most of you probably are in the realm of data science, right? So here's an example of research that we have going that really is in the realm of computational science. A common term for it is computer vision. How many of y'all have heard about computer vision or heard the term, okay? So I have my current PhD, Imtiaz Ali, who's from Pakistan. He's working on this project, certainly pushing him out of his comfort zone, okay, because he is a traditional livestock person with not as much computer training, okay, but he's taking it on and doing a great job. So we have a population been collected through a collaborative project of Hereford and Hereford cross cattle. And some of our questions surround the fact of the white pattern on their face and also the eyelid pigmentation that potentially leads to cancer eye, right, if they're more susceptible or not. But we're talking about 5,000 to 6,000 animals with pictures. Nobody wants to hand process that, okay? So we want the computer to help us. What's another problem with the person manually processing? What do you think? Error and bias, exactly. It's subjective, okay? So why not get the computer to do it, all right? And so through this process, we've been figuring out, and I'd be remiss not to point out that Dr. Haipeng Yu, who is a former master's student, went on and did his PhD at Virginia Tech, is now doing some of this stuff at his position in Florida, and he's helping us a lot. We're trying to do a three-step process. We're trying to get the computer to recognize the face part that we're interested in. We're not interested in the whole thing, okay? We're just interested in a particular region. And so we have to manually tell it, here's the region that we're interested in, okay? And then we have a set of these, and we put it in what they call a training model. We put that training model in, and then hopefully we put a validation set in to where it says, yep, I found the face without any problems, okay? Step one, check mark, right? But that's not all we want to learn from it, because we want the actual potential pattern. And so we want the computer to be able to say, well, I found the face, and based on facial characteristics that we've labeled and it trained, it can sort a handful of these into 3 categories or however many categories you have interest of, okay? And so we would go through this and do another training method where we'd say, okay, you found the face, and this face would be characterized as partial white, right, group 2. And then we would do that on a whole host of them and train so it recognizes this is a full white, this is no white, right? And it would put through that process again on ones we hadn't used, and it should group them and give them labels that we're looking for, okay? Now, why are we doing this step? No white is 0%, right? Full white is 100%. We don't need it to calculate a proportion based on that, right? But we do need to calculate a proportion of this. And so it helps the computer by knowing that, one, we found the face, we categorized it, and now we're only going to take it the next step, this partial white, so that we can look at how much of that face is white, okay? So why are we going through this step? What do you think would be a potential reason that we're trying to label these cattle faces? yes, very much so. Great point. It certainly will speed up our processing. It does depend on how much of those are actually partial versus full. We don't have a whole lot of these non-whites in our population, but certainly it would speed that up. What would be, and this is kind of just trying to see if you were getting at it. I mean, we're looking at genetic inheritance, right? And if we can provide a phenotype that would have potential genetic inheritance, we could take it to that next step and look at maybe a genome-wide association study, right? To identify if there's regions on the genome that are contributing to the fact that there's full white versus partial white versus no white, okay? Is it a major gene? Maybe. Is it multiple loci involved? Probably, okay? Yeah.
You mentioned at the start of this example, like the correlation between like depigmentation around the eye
Yep.
and cancer. So just the model was the next step and say not just percentage of white, but where white is? Is that possible?
We could, yeah. So this is just an example of capturing the face. We actually have eye pictures themselves so that we could make more precise descriptions of that eyelid percentage. We could. I would say, where would the question lead you, right? So where is the eye? And the reason behind that is if we think about genetic development, the process of placing a non-functional cell that, in other words, has the white, doesn't have the melanocyte, that would typically be random in nature during development. So I don't know if there's necessarily, there could be a genetic component that would put it specifically in that pattern. But there is genetic components that creates a similar pattern, right? And then that probably becomes some environmental cues that may alter how that gets expressed, right? Yeah, great question. So this is an example of computational science. It's a high demand of computer need, and we've still been working through, right, how can we potentially do this on that many samples, but it's a progress, and we're making it to hopefully we get a phenotype that's useful, and then we can genotype and go that next step into the actual GWAS. So another part of data science then is data analysis, right? All of you are doing data analysis, okay? To some degree, to a lot of degree, whatever it is, okay? Because any data that you collect has to be cleansed. We have to make sure that what we entered in the computer is accurate. We have to make sure that it's in a format that's usable, right? There's been a number of times that I've gone through data and I'm like, oh, I totally put that in the wrong format and now I have to go back and fix it, okay? But learning to program them becomes helpful because you can restructure your data fairly fast if you can use a computer software to do that restructuring, right? A little bit of that data management. The other part, though, in data analysis, which ties into statistics, is going to be how we actually inform ourselves from the data itself. And it goes through that initial modeling phase into potentially the interpretation of it if it's a fairly straightforward analysis. So an example that we've done in the past is when I first started at NDSU, I was really interested in cattle temperament, specifically because the way that we score cattle temperament is subjective in nature, right? Has anybody done the docility scoring on cattle before? No? Okay, so a little bit. And so I was curious, one, that whole question of what are we really selecting for on this docility, okay? But then I was also interested, are there other more objective ways that we could capture temperament based on that relationship? And so there was another faculty there at the time that she was interested in dairy cattle and dairy lameness, and she was using this scale to look at motion and the amount of weight shifts to get at lameness in the dairy cattle. And so I was like, well, let's see if that works on these calves for temperament. If they're more aggressive, more wild, they should shift more, have more changes in their weight. If they're more calm or docile, they potentially stand still, right? And the only caveat to that, if you understand temperament in any species, is that the freeze mode can offset that, right? If they're so scared they're frozen, they're not actually going to move, right? But we could still try and see what kind of relationship it had. But this scale collects data every second for however long you run it, and it captures it on 4 platforms, right? So our first task was to think, how in the world are we going to process this into a data point that's meaningful for that animal? And then how do we use that to infer our question on a multi-animal basis? Okay. So we found that we needed to first identify the starting point of each. And it's not that we didn't make a note about when the animal entered the chute. It's just that the timing of that was always offset, right? So an animal comes in the chute, and you can see behind it that it's in a typical standard chute, and it's going to balk a little bit at the space in between, okay? And so we were trying to get those calves in to where they could stand or move however they wanted to on the scale and make sure it was working. And so we had to create a programming method to actually find where the majority of the time they had at least 3 of those platforms reading some level of value, right? Indicating the animal was fully on the chute, and that we could use that as reliable information about weight shifts, okay? So this is just illustrating from that publication how we came up with that process, right? Not that you need to understand it, but just showing you that that data analysis had to happen before we could even go to that next step, okay? And all of you, especially on the graduate side, right, you're going to be doing that to be able to get to the question that you need. We did end up going on and looking at 2 versions of this. We looked at the standard deviation over 500 records, so that it was standardized across all of them that they had 500 records. And then we also looked at the CV, so the coefficient of variation for that weight shift, right? And there was a rationale behind that, and I'm not saying it was the perfect way to go. At the time, we thought it was probably better because we were concerned that the differences in the weights of the calves across the population we were working with would be too drastic, right? You have an 800-pound calf come in, its weight shifts are naturally larger because it's a heavier calf, versus a 600 or 500-pound calf, they would have naturally lighter shifts in their variation. The biggest problem with that coefficient of variation is to put it pretty close to 0, and trying modeling that becomes challenging. Okay, and so trying to think through some of those caveats is important in your data analysis phase. So data science incorporates a lot of things that we do as researchers. That computational science is a big part, but another big part that we see, and I've been watching some of the job markets, is there's a ton of data scientist posts, but they're all in one particular field. Does anybody know what field they may be in? Health sciences. Okay, we have so much human data, right? How do we process it? So a lot of that's focused there, and it's very specific to the programming and platforms that they work in health sciences. And that doesn't always translate to maybe what we're doing in agriculture, okay? And so some of the techniques they have may work, but us as agriculturists or natural resources have to think about our own domain knowledge and how we use that outcome to then apply into interpretation, okay? So one of the things I'll point out here, this is from that same temperament study. We used factor analysis to identify if there were these hidden traits, okay? So we had docility score, we had a temperament score, which is another subjective but slightly smaller scale. And we had a qualitative behavior assessment that had 12 behavioral attributes that they were assessed on. Again, subjective. And then we had our scale values. And so we were trying to understand potentially how related that scale data was to some of these attributes, and if it worked. So our first attempt at this was to look at those subjective scales, all right? And what we found was that there's 2 factors, so 2 hidden factors that connect these, okay? And that's all we could report if we wanted to, but we need to go that step further in terms of domain knowledge, right? So if I look at factor 1, and I'm looking at the darker pink indicating its positive impact on that factor, okay? I'm seeing that it's heavily loaded on difficult or aggressive type behaviors, right? And I know that from the TS only because I'm familiar with the scale, and that 5 on that scale is a highly aggressive animal. So if it's more positive on a TS, right, we're getting more aggressive, more wild in nature. But it's also supported by the fact that active is higher, relaxed is low, right? The opposite side of that. And that irritated is high, right? So instead of just saying, well, we have one hidden trait, we could actually relabel that trait to say it's an animal that is going to be more difficult to handle based on the scores that it received. And then we could potentially take that to a predictive mode, right, if we wanted to. On the other side, the other hidden variable you'll see starts looking at easy or calm attributes, right? That it's more less energetic, I guess I would say, in that capacity for the same reasons, more positively loaded in those positive or less aggressive tendencies. Okay? So another big part of data science then is making sure we have appropriate domain knowledge and how to incorporate that. And I think that's a big gap in our industry, right? Is being able to take that domain knowledge and know how to do all the computer stuff and get the questions answered the way that we want. Any thoughts on that before I go to this part? So biostatistics really is not much different in the capacity of statistics. It's just the actual application to our biological sciences, right? And that can be anything from agriculture to wildlife, natural resources, right? Its origin actually was influenced a lot in the field of genetics because of the individuals that were trying to understand both Mendelian inheritance, so the pea plant experiment, right, if you think back to that, and then also how we see some of these traits that are more quantitative in nature. And back then they didn't really understand the term quantitative, but they were seeing that observation that it was more continuous in nature. And so biometry or biostatistics really got its roots there and has evolved a lot. The problem, and I'm glad to see that a lot of you recognize you probably have messy data, is that even though we have really structured experimental designs, a lot of times the data is just messy, okay? And we have to still take those biostatistics and our knowledge of how that statistical process works so that we can infer what's feasible out of that data, okay? So the example that I have here is from a recent publication, and one, I think it's cool to do a 3D graph now, right? I had never done a 3D graph, but I thought it was appropriate in this case. And so what we're looking at is a population genetics study. Some of you heard me talk about cow size in the master's thing. So we took that population and we wanted to understand the admixture because there's this interest. You see it on the cat and dog side. You can send your DNA sample in and they'll tell you the percent of each breed that dog has, okay? And I'm like, well, there's talk of that in cattle. Can we actually do it? Okay. And another part of this was a twofold approach. We knew that we were wanting to do more complicated questions with this population. And if we have a very admixed population, that was going to make it hard to ask those questions unless maybe we could piece apart that breed composition. So luckily, that center really recorded the pedigree very well. So I knew based on pedigree the expected proportion of the different breeds those cows and heifers had. And we also pulled in the purebred bulls that were used to produce a lot of these heifers. And we also used an online database of genotyped purebreds. And we went through two-step process. We went through a population genetic studies just using purebreds, which are going to be up here on the top. And then we went through the process of looking at if we put the admixed individuals, so those crossbred individuals, into the analysis, what did it do, right? Could we still differentiate different populations well enough? And if we could do this part, then we probably could understand actual percentages based on their genomics. The purebreds look really pretty, don't they? Okay, color scheme here. The red over there is American Aberdeen. That was an Australian-based offshoot of Angus. Selected highly for growth rate. And so it would have slightly, the expectation is that it should be slightly off from the Angus and such. And we see that in the different axes of that graph. It's pulled away, right? The green are going to be our British type breeds. So Angus, Red Angus, Hereford, Shorthorn that we had in the population. And then the gold are going to be our continental breeds, Limousin, Shorthorn, and Gelbvieh, okay? Simmental, sorry, not Shorthorn. So you can see the populations clearly pulled apart, okay? There's some overlap of a few of those that I may or may not have expected, like Hereford and Shorthorn are fairly close to Gelbvieh and Simmental, right? But then when we put the admixed individuals in, it's really hard to pull them apart, isn't it? You can see a bunch of colors meshed on top of each other, and it's not clear if those are one particular breed or not. And I've done this graph in multiple colors so that I can try to see what's which population, and it's just messy, okay? So what could we infer from this, right? It takes a little domain knowledge to fully piece out what could be going on, and I don't even know if it's really the only thing. But I started seeing it here. Okay, again, we had a random sample that were from this database, so I have no idea their origins except the fact that they are purebreds because they were deposited that way. And then, of course, we have the purebreds that were involved in the population that influenced these admixed. Okay, so in cattle, there are several breed associations that allow grading up, right? And that is a process where generationally you're trying to increase the percentage of that breed in their offspring so that over time you get an offspring that is a high enough fraction they consider them to be purebred or close enough. Okay? But if we know anything about genetic inheritance, that doesn't mean that they're actually getting all of those genes every generation, right? And so what we're seeing is there's enough admixture along with that grading up for those breeds that we're losing the distinctness of those breeds as we mix them, right? And so it's probably becoming to a point that it's almost impossible to do a analysis like you see in cats and dogs, right? We can't piece them apart on an actual percentage and know that we're going to get a reliable percentage out. Certainly there's probably other factors influencing there, but to me that's probably a very big one, the grading up process and how you're allowing some of these genetics from the different breeds that once stood on their own be introgressed into these other breeds. Okay? So we should expect then, as both data scientists and biostatisticians, that we're going to run into instances that they aren't going to make sense, and we have to try to figure out how to explain them if we can. So the title, which I thought after I wrote it and sent it off was quite lofty, I just want to spend a moment on sustainability, right? And honestly, it's pretty debatable how you define sustainability. You could think about it in environmental or economic or social impact. We're all kind of interested in ag and natural resources, how this is going to continue, right? If we're continually growing as a human race, how do we continually produce the food or preserve our resources? And so it kind of goes back to this concept of what's the best fit and how do we make sure long term that we're keeping that in mind, right? So I brought up this in the master's class, and really it was talking about sustainability. If that production system can handle moderate to larger size females, and it's not going to be detrimental to the resources over years, then that's okay, right? But if we can't, then we need to consider making sure we're watching those size of animals and being mindful of the impact on those resources before we put more animals out. I think this translates to a lot of different things. I just wanted to provide kind of a specific example. So to you, right, we are amassing amount of data. There's lots of technologies that are coming out that are allowing us to capture all of this information, but then how do we use it, right? And in the ag and natural resource side of things, we're not going to get any more land, we're not going to get any more natural resources unless we do efforts to sustain those. So we need to use the data to our best ability. One of the things I point out here with this publication, it's a recent publication that we did, and it actually looked at ewe litter birth size and how that may impact their longevity, right? And out there, one of their big culling criteria is that by the age of 7, that ewe is going away. Because this is out in Idaho on some really harsh rangeland, okay? And so their teeth are just worn down. They can't handle eating out there past that age, and the only reason that they would do that for research purposes, okay? And so in that case, they wanted to know, right, well, we want them to stay, we want to get the profit from these ewes, but we have this caveat that they're cold, right? So are there other factors that can help us extend their life? And we started looking at that, and one particular was the birth litter size. And we do see those that are born in more like single or twin bases will do better than those that are born as triplets. And there's a slight favoring of singles over twins, okay? But this is a litter-bearing species, okay? And so the concept in the pull there was more producers need to be mindful that if they are selecting for better fertility, there is too much fertility, right? That you don't need to push towards. You need to find a balance and note that your average should somewhere probably be around a twin, but it shouldn't necessarily be towards a triplet, okay? The other thing with this is that I don't have this published yet, but we have done a study where we included dominance effects, okay? So this is a study where we're looking just at additive genetics, so how alleles are summing across, but we wanted to know dominance is sometimes important, right? The black allele overrides the red allele. That's probably happening in reproduction. So what can that tell us, okay? And that study has really shown that the heritability of longevity skyrockets, okay, because there's huge dominance effects. And I bring this up on here because this was a massive amount of data we were dealing with. It was from 1950 to 2015, and they've continually collected the data on thousands of ewes, okay? So trying to process that and get informed decisions from it. I'm just a normal animal scientist, right? But I have to work with data sets like this if I want to answer those questions, and it helps with our resources because we're not just doing a project and knowing that we may or may not have power, right? In this example, we certainly could have power, and we probably could have some really cool questions answered with it. So having to step through that process of learning more about the computational side is helpful. All right, so the last thing I'll leave you with here is another recent project. It's almost a submission stage. And this kind of drives home that point of what are we actually collecting and is it useful? And so on these heifers that we had, we collected a ton of data on them. I mean, I can't even sit here and list the amount of data that we collected. And we wanted to understand what underlying traits could they all be really representing. Was it just one? Was it multiples? How related were they? Okay. And so we went through this process called factor analysis, and we used an exploratory method to see what hidden variables could be there. And then we said, okay, well, based on what we found, let's confirm that that's really there with the data. And what we came out with is that there's really just two primary ones, okay? There are some relationships to this other one, but really what we're looking at is size in this top part and carcass characteristics or yield grade, okay? I also found pretty interesting that IMF, which is intramuscular fat, stood on its own, and it did that multiple times. So there's something about IMF that's distinctly different than yield grade and some of those other carcass size characteristics. The point here is that we're now understanding the data better, that really there's just one or two traits of importance, and that we could look at understanding their relationship through a Bayesian network learning, and that may lead to a better statistical model for our questions, right? And so that's something you may need to consider as well, is that if you go down the road of having all of this data, how related are they? Are they leading to a similar type attribute? And then what does that actually tell us about the question we're trying to answer? Okay. All right. Any discussion? So what kind of questions or discussion do you want? Oh, yes.
Can I go to your previous slide about the youth project?
Yeah.
So I have a question regarding that. If this type of analysis where you say you have like multiple years, right? You said like 2009 to 2014 or something like that.
1950 to 2015.
So
Yeah.
even more. So how do you take into consideration the location, the environment these animals are in, the breed, the age of these animals?
Yeah.
Right, like, 'cause it sounds very interesting, but at the end, is this result, does it apply to everything or just to a specific population?
Yes. So, and that was more of me just thinking about my time frame, not going too deep into the structure of the data. So I appreciate you asking that question. This is all from the USDA ARS sheep station in Idaho. They keep very detailed records. All right, and they've actually developed several of the breeds that a lot of the Western U.S. use nowadays. Okay, so like Targhee and Columbia, and so they have. We took the entire data set And we narrowed it down to their 4 major breeds. So one, there was enough sample size per breed that we could put that breed effect in the model, okay? And I'm talking thousands of records on each breed. And then we also looked at the data in the sense of a yearly basis, okay? And although we had access to 1950 to 2015, we actually cut it down to around 1986 because there was a huge change in their management structure around that time. And we decided that although we had access to those 1950 type ewes, it wasn't going to tell us a whole lot about the current ewes that were going on. And so we did some filtering that way. And also we looked at litter sizes within the populations of breeds, right, to make sure that we weren't going to bias in those questions. And we had to play a lot with the statistics, trying to see what kind of counts per interaction could be there that we could ask a really good question or not with. And then go from there, right? I think that's kind of addressing. Is there another detail that you wanted to ask related to that, Julie?
I'm just interested about what were the major findings of that research? Like what did you find about
So,
the whole year?
oh, yep. So we found that leaning towards the twin to single side tended to elongate their life in the production system. Yep. Now, there was that kind of comment too, right, that it's not that we need to get rid of twins, but we need producers to be aware that moving towards triplets is probably not ideal, right? That you need to mitigate that. Now, this has to deal with a lot with the western range, and I wouldn't say that that necessarily translates to a eastern or Midwest setting where you have high forage production and it's not as browse oriented, right? So out in the ARS station, it's high browse. And that would tear down their teeth a lot faster, right? Yep.
I'm wondering how much is this related with the placental functional capacity, right? Because I see bananas mainly there and Reynolds, they work with intrauterine growth restriction. So how is my ewes that are in utero experiencing more intrauterine growth restriction because they are triplets instead of singletons or twins? So how is this could be the trigger factor of my ewes not having these successful reproductive traits later?
It could be. There's a lot of questions you could try to piece apart there. So the second step to this, I indicated we looked at dominance. We also factored in birth litter size. So trying to understand a little bit of that capacity of how the size that they were born and influential on their longevity in that capacity. Because that would say something about thriftiness and their ability to move on into a productive state. Birth weight was not important. It hardly counted any kind of regression coefficient with it, and it was not significant. So there could be other factors post-birth that becomes an important factor in their longevity, right? Not necessarily a uterus type thing. I wouldn't say I would rule it out. I just think there's probably a post-birth type situation, environmental aspect, that then drives their ability to go on. The other part that we did in the second publication is age of dam, right? To see if those that were born to really young dams, right, change things. And I'm drawing a blank right now on that statistical outcome, so I don't want to say anything, but but we did look at that part in relative to see if that helped answer some of that question too, right? Yeah.
My other two things, I'm sorry, and this is just interesting.
Yeah.
But the other two things that I would think about is the sire as well of these
Yes.
animals. Do you have that in record for this study?
So this was done using an animal model, so we used pedigree and
Random.
yep, and so treated those animals as random with their pedigree, knowing their additive relationship. And then the second step with the dominance would include both additive and dominance, inheritance based on familial relationship. Mm-hmm. Yeah.
you.
Yeah.
Okay, I'm gonna try to, you know how in disciplines you've got different words?
Yeah. Yeah.
So, can I make an attempt to try to restate to you some of what you've been saying about the data science and everything, sort of in the words that I use and that my students are used to me saying?
Yeah.
And you tell me if I'm thinking about this wrong. It also is based on the way that somebody has defined data science to me. So here I go. So we have these big data sets,
Yeah.
and they're either over multiple years, and they include a lot of confounding variables. So being all of those different things. And obviously in biological systems, you've got like 9 million confounding variables. And the longer the data set, time-wise, temporally, and spatially, the more that adds up, right?
Yep.
And so the idea behind data science, and I guess tell me if I'm incorrect, if I'm thinking about this wrong, but the idea about it is to go in and to look at that data set and identify patterns based on the data itself, but also the biological meaning of it.
Yeah.
And identifying those connections amongst that data such that you could potentially simplify a statistical analysis later.
Yes. So there's two parts to that really, because you're right, confounding variables can happen. It's the level of confounding we need to first be concerned with.
Yeah.
So if we have something that is completely identical to another term, it's just another way to represent the data. It could mean something completely different biologically, but there's no way for us to piece apart, right? So when we think about the different ways that we can approach that statistically and with computational science, it becomes what are those traits that have, even if they are slightly confounded, they're distinct enough that we can ask relationship questions about, okay? And so if we go back to this example of the, too far. Oh, I was at the end and I forgot. Okay. So if we go back here, we looked at different things like mid-girth and front girth, right? They're all girth measurements, but they're different parts of the body. And when we looked at just simple correlations, they stood on their own enough that we knew they were related because they were from the same animal, but they said slightly different things.
Right, from the data side.
From the data side of it. So so go ahead.
Yeah, no, so it is. So it's looking at these different things and first of all asking whether or not that confounding variable actually is doing anything, or if it means something, meaning that it's correlated with another variable such that you can ditch it.
Yes.
Right? But it's kind of the pre-side of it, going through and looking at that data and everything before you really run that, do the model selection side of it or something like that.
Yes.
Okay.
So all of this right here was a step so that we could actually do GWAS with the
Yeah,
structured
yeah.
equation modeling, right? So we haven't even done the GWAS at this stage.
And I think that a lot of times for students and stuff, a lot of what is left out of their education in statistics is this data management, data science side of it.
Yeah, I agree. I don't think anybody ever used the term data science with me when I was going through grad school, but I
Yeah.
essentially was doing data science.
Yeah,
And-
I call it data management.
Yeah.
I kind of mean the same thing.
I do agree data management is there. When I think about data management, I sometimes think about relational databases, and I would not say that I'm strong in that type of situation. I understand what relationship databases are, but actually creating one, I'm not sure I would necessarily do a great job.
I include the relational database part of it
Yeah.
as more of the data entry, data design
Yeah, okay.
term.
Yeah, I would see it that way too. Yeah. Sorry.
Okay, just wanted to make sure it
Fun-
translated into my
yeah.
language.
Yeah, so I had a fun conversation. I see yours just a second. Fun conversation because I worked with somebody in biological sciences and they were talking about plasticity, and I was like, what in the world are you talking about? And it turns out it's not quite the same thing, but it's very similar to G by E, right? Genotype by environment interactions.
Yes.
And I'm like,
Yeah.
okay, I know what you're talking about sort of now.
Yeah.
What's your question?
So I just want to follow on the previous question. We're dealing with big data sets, complex observations and characteristics, often correlated responses. So my question is, how do you know, or how do you make sure that your cause-effect relationships are accurate?
Yes, that's an excellent question. Some of that comes from a domain knowledge standpoint, right? If we don't understand the organism or what we're trying to answer. The other part of that, though, I think is a sound understanding of statistics, because we can try to ask questions and it could tell us that it's significant, but it may just simply be an artifact of size, right? Something is driving it because of the sampling that you did. And so being willing to ask those questions to say, is this real? And how can I trace down if it's real or not? Getting the help that you need to guide through that, especially for grad students, right? I think that's really important.
Maybe that's where repeatability comes in. If you repeatedly get the same outcome.
Yes, yes. And I think that drives, I don't have those points here. With this data set on this last slide, we actually approached this two different ways because the sample size was fairly small. And we wanted to see, okay, if we just let the data speak, what would it say? And then we said, okay, we know we have a larger sample size of this other data compared to the carcass. So if we let them drive separately, what happens? And we see that we still had the power within that smaller data set to see some relationships that got lost with that separation, okay? And those relationships made sense biologically, right? And then we can take it a step further, and we've done some preliminary GWAS on that. I just didn't have it in a state that I thought was good enough to present. But what was interesting, it didn't matter which version and whether we did body size structural equation modeling with that relationship to body composition, all these same genomic regions kept popping up, right? And so I looked at my PhD student who was working on this, and I was like, guess what you get to do? You get to go run this trait, and this trait all by themselves and see what we get for the GWAS, right? Because I was curious whether it was just really those traits driving it or if it was really something underlying that kind of thing. But it's an excellent question to ask, right? Good, yeah.
Is there any specific software that you find is the most friendly or amenable to dealing with large data, the data science side of it?
I love
Like
R. Yeah, I love R.
Okay, good, I like that.
I do think when it comes to the machine learning side, if you want to dabble in that, you're going to have to move into Python.
Yeah,
Jupyter Notebook is a really nice, handy tool, kind of similar to RStudio. Makes it a little more user-friendly. So Jupyter Notebook is a good place to start with that Python. But yeah, if you're going to move into more of that machine learning, artificial intelligence, you're probably going to have to move into Python. I don't know I haven't looked too much what's set up in R for that. I think there are capabilities. I just think what's usually pushed in the realm is that Python is better suited to that analysis. So, yeah. Well, I really appreciate the engagement this afternoon, and I don't know if we still have time, Dr. Leatherwood, or not, but okay. I'd also like to just thank y'all for letting me come and speak. It's been a lot of fun to meet everybody and get a chance to interact on this. So.